we're going to do today is finish up the review of some of the key things from self-phys, focusing on muscle contraction, cardiac, and then smoothing cardiac, and then smooth muscle contraction, and then start with an overview of the cardiovascular system. So we're moving into our first organ system, really, so this class now starts in a way, not much review, although of course there will be bits from self-phys that we'll see over and over and over again. So you should all have picked up a key concepts document. These are the list of things to understand, to know for this section of the course. Cardiac anatomy, vascular anatomy, mostly from your coloring book, and the physiology that we'll be talking about. The anatomy will start in lab today, so this afternoon and this evening we'll do heart anatomy, human and pig heart anatomy. Hopefully there will be time to, if you have any questions about the problem set that you may have started working on. That's due on Sunday. We can discuss those today as well. So we're going to be starting with skeletal muscle, but using that as a springboard to understanding cardiac and smooth muscle. So here is the slide that we ended with on Tuesday. I'm of two minds about whether to go through it again. The main points, let me not go through it all, but get some of the main points. The main point is that the dissociation of ADP and phosphate is what triggers the so-called power stroke that allows the myosin head group that is connected to the actin-thin filament to pull on the actin-thin filament. Then binding of ATP is what releases the actin head, the myosin head from actin. Let me do that again because I screwed it up. ATP binding to myosin head causes it to release the actin. Hydrolysis of ATP puts it back into the extended conformation where as long as calcium is present, it can bind to actin again and do this over and over and over again, millions of times, to pull the Z disc closer to the M line, closer to the middle, shortening the sarcomere. So you should be able to walk through this sequence of events and tell me the story of muscle contraction. This is true for skeletal, cardiac, and smooth muscle. Just to give you a taste of where we're going, taste might not be the best word. So here is a video of a heart undergoing contraction and the so-called Wigger's diagram that we will get to before the end of this section of the course. So before the end of this section of the course, you will be able to understand the sequence of events that leads to this heartbeat and generates this set of curves. That is where we're going. That's the goal for the next couple weeks. And you will eventually, so what are we seeing in this image of the heart? Well, we're seeing muscle contraction. These are the ventricles contracting. Here is the right atrium and the left atrium contracting. We're seeing valves opening and closing, opening and closing, opening and closing, a valve closing and opening, closing and opening. So we're seeing valves opening and closing, we're seeing muscle contraction. You're going to learn about the anatomy, the names of these structures in the anatomy part of the next few days. And we'll see this, not beating, we'll see this in lab today. That's why this is really why we're doing this. Actually, I'll stop it from beating. Okay? So So skeletal muscle has that mechanism of contraction that I just talked about. Skeletal muscle is an interesting, the cells of skeletal muscle are interesting because they are huge. So one skeletal muscle fiber, one skeletal muscle cell in my biceps can be, inches long. It can reach from my elbow to my shoulder. One cell. It's not very big in diameter, but one cell can be very long and they are formed by the fusion of precursor cells, hundreds or thousands of precursor cells that are cellular in diameter, in size, tiny microns, fused together to form a cell that is inches, feet long in skeletal muscle. That doesn't happen in cardiac muscle. In cardiac muscle, the cells are cellular in size, tens of microns in diameter, in size, length and diameter. They don't fuse. So here is an image of cardiac muscle. Here is a cartoon of cardiac muscle. So what is similar between cardiac muscle and skeletal muscle? And what is distant? What are some of the similarities and dissimilarities? That's an important thing to think about. Well, the cells are not formed as fusion of precursor cells. The cardiac muscle cells are individual cells. They each have a nucleus. They each have a plasma membrane. They have sarcomeres. So here's one sarcomere. Here's another sarcomere. Here's another sarcomere. We can see them also in the micrograph, but I can't reach that high. So they have the contractile mechanism for getting shorter, just like in skeletal muscle. They have mitochondria to make ATP, abundant mitochondria to make ATP, because the muscles are contracting all the time. Every second your heart contracts. They have they have a special structure that connects one cell to the next cell. So each cardiac muscle cell is a cell, but it is physically and electrically, there's the key thing, physically and electrically connected to its neighbors. The physical connection is called the intercalated disc, and the electrical connections are called gap junctions. These are channels. These are ion channels. You remember those from the last week and from self-phys. These are ion channels that actually connect the cytoplasm of one cell to the cytoplasm of the neighboring cell. Electrical connection from one cell to the next. Not from one cell to its extracellular fluid, but from one cell to the next cell. The gap junctions are two membrane ion channels head to head. So if one cell fires an action potential, depolarizes, undergoes all kinds of changes, the next cell gets the signal through these electrical connections. So that is a dissimilarity from skeletal muscle. Skeletal muscles don't do that. Another dissimilarity is that each skeletal muscle fiber, each skeletal muscle cell is innervated by a motor neuron. Heart muscle cells are not innervated by motor neurons. They are innervated by sympathetic neurons, and we'll get to that later. Let me just point out, you know that when you're stressed, your heart beats faster and stronger. Whether the stress is psychological, because I'm going to remind you that there's an exam in a couple of weeks, you just got a stress stimulus, or a stress stimulus because you decided to take a run and your body is stressing your heart. That is neurophysiological. The nervous system, the sympathetic nervous system, tells the heart beat stronger and faster, but it doesn't tell the heart to beat. The beating of the heart is intrinsic to the heart. The rate and strength of the heart is controlled by the nervous system, but the beating of the heart is intrinsic to the heart. And we'll talk about pacemaking next week. We'll talk about how the heart sets the pace next week. Anyway, so right now, what you need to know is that each cell is not innervated, but it is connected to its neighbors. So if one cell fires an action potential, the next cell depolarizes and fires an action potential. Cardiac muscle and skeletal muscle are similar in that they both have T tubules and they both have the sarcoplasmic reticulum, the model, which we can look at in lab today if you want. I didn't bring it here. Remember the network of T tubules and the network of sarcoplasmic reticulum wrapping around the sarcomeres of thick and thin filaments. You saw that three-dimensional model. That's happening in cardiac muscle also.
You said they both have what?
They both skeletal and cardiac muscle have T-tubular networks and sarcoplasmic reticulum networks surrounding sarcomeres. So, just like in skeletal muscle, when an action potential propagates in the surface membrane, it causes the release of calcium from the SR, which is what triggers contraction, which we've already covered. In skeletal muscle, the thing that senses the voltage in the T-tubule, the thing that senses the action potential in the T-tubule, and tells the sarcoplasmic reticulum to release calcium, is a physical connection. It's a physical connection between something called the dihydropyridine receptor, which is the voltage sensor, and the calcium release channel, which is the route by which calcium is released from the SR. That's a physical connection in skeletal muscle. In cardiac muscle, it's different. So here's a difference between cardiac and skeletal muscle. Skeletal muscle has a physical connection, which is cartooned here, between the voltage sensor in the T-tubular membrane and the calcium release channel in the sarcoplasmic reticulum membrane. Is that cartoon clear? So it's blowing up this connection between T-tubule and sarcoplasmic reticulum membrane. So this is T-tubular membrane, right there. This is SR membrane, right there. And this is the connection between the protein that senses the voltage in the T-tubule and tells the calcium release channel to open and release calcium. In skeletal muscle, it's a physical connection. In heart, it's different. In cardiac muscle, there is no physical connection, so this cartoon is wrong for heart. What happens is the dihydropyridine receptor is, in fact, a calcium channel, and calcium flows in through the dihydropyridine receptor. So it flows from the extracellular space, right? This is extracellular, this is extracellular, this is where calcium is relatively high. The dihydropyridine receptor senses the voltage and opens a channel, a calcium channel. And calcium flows in through the surface membrane. So that's one way in which calcium goes up in the cytoplasm, about 10%. So you're saying, okay, 10% is not very much. Where's the rest of the calcium? The rest of the calcium is caused by something called calcium-induced calcium release. So in heart, the calcium that comes in through the dihydropyridine receptor voltage-sensitive ion channel, that calcium causes the calcium release channel to open. And so 90% of the calcium signal in heart comes from the SR, which was triggered to open by calcium-induced calcium release. So are the similarities and dissimilarities between heart and skeletal muscle clear? This is the mechanism by which heart muscle gets the signal, the calcium signal, to contract, which is after all what causes the heartbeat. Happy to entertain questions, because this cartoon is of skeletal muscle. Cardiac muscle is similar in many ways. It's got a T2 wheel, it's got an SR, it's got a calcium release channel, it's got a DHP receptor, but it doesn't have the physical connection. It has a calcium-induced calcium release connection. Is that clear? I want to make sure it's clear. Well, we can, if you process this over lunch and you want to, or dinner and you want to talk about it in the lab, you can do that. So, calcium triggers contraction. Contraction is a shortening of sarcomeres. That's all that this slide is supposed to say. You know that. Shortening sarcomeres is what causes the muscle fibers to get shorter, and if a bunch of muscle fibers get shorter, the heart gets smaller and contracts. Okay, so let's do a clicker. See if this is connected. Which of the following is required for myosin to bind tightly to actin? What do you need inside the cytoplasm for myosin to bind to actin? Just binding. We're not talking about contraction. Just binding. So let me go backwards. What do we need for myosin to bind tightly to actin? I'm seeing many partial correct answers and a few completely correct answers. What do we need for myosin to bind tightly to actin? We need calcium and ADP. Is that clear? All right, go back to the cartoon. We need calcium to move troponin and tropomyosin out of the way. And we need ADP to be bound to myosin. And then we get this high affinity binding of myosin to actin, which was the question. Okay? Let's do another one. Which of the following is required for the power stroke? So which of the following is required for the conformational change, the protein conformational change that we call the power stroke of muscle contraction? Okay, good. I'm seeing more fully correct answers. So this one is more straightforward. This one is B. You need ADP and phosphate to dissociate. That's what causes the power stroke. And you should be able to go through, like this clicker will be available on Moodle after class. And you should be able to say, okay, what happens? So this is what happens in order to trigger the power stroke. What happens here? When this happens, what happens? When ATP, for example, when ATP binds to myosin, what happens? When ATP is hydrolyzed by myosin, what happens? Because I could write another exam question that was just like this, except for which of the following is required for the myosin to undergo the conformational change from angled to extended. That's a question. Let's do that as a clicker. What is the conformational change required for myosin to undergo the conformational change from, they call this cocked, angled to extended? Good. Many of you, I'll give you a minute for those of you who haven't pulled this up right away. But the conformational change from cocked to extended is caused by ATP hydrolysis. So that would be easy. Questions about cardiac muscle? Okay, let's go on to smooth muscle. We can do this relatively quickly. Smooth muscle is the muscle you're not very aware of because you have hardly any conscious control of smooth muscle. These are the muscles that line the walls of various internal compartments, like your digestive tract is lined with smooth muscle. And we'll talk about that when we get to the digestive system. Your urinary bladder is lined with smooth muscle, and you don't have control of it. Well, you have limited control of it. The sphincters in your body, so for example, the sphincter that controls whether you're urinating or not, you do have control over. You've learned how to control the sphincter, the smooth muscle there. So there are some that you have control over. Infants don't have control over it. Right? That's something you learned how to do. The point is that smooth muscle lines these tubes and these sphincters, things that close or open in your body. That's what smooth muscle does for us. And so the arteries and the veins that we're going to be talking about real soon are all, many of them are lined with smooth muscle. And the smooth muscle controls the diameter. A relaxed smooth muscle allows the diameter to increase. This is key, and it's not on the slide. A relaxed smooth muscle of at least arteries and veins allows the artery and vein diameter to increase. A contracted smooth muscle causes the diameter to decrease. So what are some of the similarities and differences between smooth and cardiac and skeletal muscle? Smooth muscle does not have sarcomeres. They don't have T-tubules. They're not striped. So what does striated mean? Striped. Striated means they're striped. So where do the stripes come from? The stripes come from the fact that they're aligned in sarcomeres. Striated just means striped. So cardiac muscle, let me just, going back to when cardiac muscle looks striped because it has well-defined sarcomeres. And so they call it striated muscle. Skeletal muscle and cardiac muscle are striated. Smooth muscle is not striated, Doesn't have sarcomeres, it doesn't have T-tubules. Most of it is not under your voluntary control. If I say to you, I want you all to increase your blood pressure by 10 millimeters of mercury, you can't do it. You don't have that control. They are connected by gap junctions. So smooth muscle cells are connected to their neighbors by gap junctions similar to cardiac muscle. So that's a similarity between cardiac and smooth muscle. All the others are differences. Well, do you have control over your cardiac muscle? No, you don't have conscious control over your cardiac muscle either. You can't say, I want my heart to beat faster. You can't just tell your heart to beat faster. You have to do something like, I have to imagine that Bob is going to give me a quiz right now and I wasn't prepared for it. That could get your heart beating faster, that imagining that stress. Many of you who are practiced in any form of meditation can slow your heart rate. If you just meditate, you can slow your heart rate. So you have control over the rate, but you can't say, I want my heart to stop for a minute. You can't do that. Smooth muscle lines, so for cardiovascular, smooth muscle lines the arteries and veins. It lines other tubes in your body too, which we'll get to. There is longitudinal smooth muscle and circular smooth muscle. So if this is a tube, use my limited artistic skills. So here is a tube. Let's just say an artery, small artery. We have smooth muscle that is organized longitudinally. Along the smooth muscle. And we have smooth muscle that is organized circularly, wrapping around the smooth muscle. Poor diagram, but you get the idea, right? This is supposed to be a smooth muscle that's wrapping around. This is a smooth muscle that's, so for example, here is a smooth muscle on top of that tube. And here's a smooth muscle wrapping around the tube. Circular and longitudinal smooth muscle. Which type of those muscles do you think is better at controlling the diameter of a blood vessel? The circular. But they both exist. They don't have T-tubules, they don't have sarcomeres, but they do have thick and thin filaments. They do have myosin thick filaments and actin thin filaments. They just aren't organized into sarcomeres. And they're organized in this sort of meshwork of thin and thick filaments. And the network, the thin filaments are connected not to Z discs, but to something called dense bodies on the surface membrane. So the thin filaments are connected to the membrane. The thick filaments are not, but they are intercalated between the thin filaments. And so if we have myosin pulling on actin, we pull these, instead of pulling two Z discs together in a sarcomere, we pull these two dense bodies together, and we can change the shape of the smooth muscle. And if this is circular muscle around a, it's relaxed around the diameter of a blood vessel, and it goes from this to this, what happens to the diameter of the blood vessel? It goes from here, if a smooth muscle, let's just imagine, this is a circular smooth muscle. And it goes from this shape to this shape due to contraction. What happens to the diameter of the smooth muscle? It contracts. It gets smaller. The diameter gets smaller. What controls the contraction of smooth muscle? One word. Begins with a C. Calcium. What are ways in which calcium can be, what are two mechanisms by which cytoplasmic calcium can be increased in smooth muscle? I heard somebody say something. I want to get a conversation going here. Yeah, Josh?
That exchanger.
NCX. NCX, you could defend that answer. I won't ask you to defend that answer. NCX is usually used to pump calcium out, not to let calcium in. But you could defend that answer. It's just not the best answer. But Josh is on the right track. What is a mechanism? We're talking about a protein mechanism that allows calcium to increase in smooth muscle. Kendra. GQ, coupled receptors. GQ, that's what's going on here. GQ, this increase, you cytosalicalcium increased, and now you see some of the results of that, right? Binding to calmodulin, da-da-da-da-da. GQ coupled receptors can cause an increase in cytoplasmic calcium. So a hormone or a neurotransmitter that activates GQ coupled receptors can cause the contraction of smooth muscle. That is a key point. What's another way, another mechanism? Ion channels, a specific ion channel. A calcium channel, a voltage-gated calcium channel, can also trigger enough calcium to cause the smooth muscle to contract. Question?
For the GQ, is it fast acting?
It's fast enough. Smooth muscle contraction is pretty slow. And GQ coupled receptors are, that calcium signal is fast enough. So calcium is the trigger for contraction. Calcium is the trigger for contraction. We've just gone upstream from this, right? What are some of the ways that calcium can get there? Voltage-gated calcium channels or a GQ coupled receptor. That's what triggered cytosolic calcium to go up. Binds to calmodulin. Now we're talking specifically smooth muscle. Binds to calmodulin, does not bind to PKC. We don't have to think about the PKC story here. We're thinking about the calmodulin story, which is why I asked you to think about calmodulin in the problem set. Calmodulin activates a kinase. The kinase phosphorylates myosin. The name of the kinase is myosin kinase. Well, it's actually myosin light chain kinase, but the light chain is just you can put that in parentheses. Myosin light chain kinase. Myosin kinase phosphorylates myosin, and in smooth muscle, the phosphorylated myosin is what binds to actin. So the calcium and the phosphorylated myosin bind to actin, and now we do the cross-bridge cycling and the contraction of smooth muscle, turning it from this to this. And like with everything else, if it goes up, it has to go down. And here's the way that phosphorylation of myosin goes down in order to cause the transition from this to this, like after you have this stress response. You want your smooth muscle to relax again. Well, the relaxation happens because the kinase is opposed by a phosphatase. What opposes the calcium signal? What would reduce the calcium signal in the first place? Josh?
Non-nucleotide exchange.
NCX?
NCX.
NCX is one of the things that would decrease intracellular calcium, cytoplasmic calcium. What are two more things? 8 letters altogether that would cause a decrease. Brittany?
PMCA.
PMCA, and, oh, it's 9 letters. PMCA and CIRCA, the pumps. Okay, I'm trying to bring this together. Let's take a break now, because I'm kind of done with the review section. I mean, the specific review, although smooth and cardiac muscle was new, but there was a lot of new, but there was some review. Now we're gonna transition into all new cardiac and cardiovascular anatomy and physiology. So let's take a break now. On question 1, I want to remind you that there are some statements in that sequence of statements that are wrong and need to be crossed out. So it means, so if you see something and it just says, wait a second, I didn't know that happened, well, maybe you're right, it doesn't happen, cross it out, okay? In question 3, I'm asking you to tell me the story that leads to both PKC and calmodulin kinase. So I'm not asking you the smooth muscle story that we just did now, because the smooth muscle story focuses on calmodulin only. But this question is asking you to think more broadly. And finally, there is no question 5. That was my mistake. I do want to point out that, to remind you, that there are assignments in the coloring book that inform you about the anatomy. So for example, if you did the coloring book assignment for today already, especially page 104, which is coloring the anatomy of the heart, you will be better prepared for what we're about to do in class and better prepared for what we're going to do this afternoon in lab. So I just want to point out that I try to time it well. There's the operational word. I try to time it well, that the coloring assignments are actually informative or preparative for what we do in class and in lab. So my recommendation is do the coloring book assignments before class and before that lab, especially if they're on Thursday. I don't promise that there won't be some times where the coloring book is either the coloring book assignment is a little too early or maybe a little too late because the timing of lab gets complicated because things happen, some things happen fast, some things happen slow. It's a little bit out of my control, but that I try to align in time the coloring book assignment with what we're going to do in class or in lab. And that's why it's in your interest to do them before the date of the assignment on the schedule. All right. So, cardiovascular system. We're going to focus on the heart first. So cardiac, and then vascular. You know you only have one heart. But we talk about it in terms of 2 hearts. We talk about it in terms of a left heart and a right heart. So, and we actually talk about the circulation in terms of left and right. So, now what does that mean? How can we have a left heart and a right heart, but we only have one heart? Well, what we have is we have a left atrium and ventricle and a right atrium and ventricle. Our heart, which is one pump, is really 2 pumps working simultaneously in series. There's the left pump and the right pump, so we'll be using that information. We have atria, so you know that a building has an atrium. Like CST has that entry space with the sort of big high ceilings. There's an atrium. The heart has an atrium, has 2 atrium. Heart has 2 atria, 1 left atrium and 1 right atrium. Atrium is the singular, atria is the plural. So if you talk about the left atria, you're talking about more than one person. If you talk about the atria of one person, you're talking about the left and the right atria. I won't take off points if you make a grammatical error like this, but I just want to be clear about what's the difference between an atrium, singular, and an atria, plural. You have a left and a right atrium. You have a left and a right atria, and you have a left and a right ventricle. Left and right ventricles. This one's easier. The right heart, the right ventricle, is connected to the pulmonary circulation. Where does the pulmonary circulation go? Lungs. Pulmonary is referring to lungs. So the right heart is what pumps the blood to the lungs. The left ventricle is connected to the systemic circulation. What do I mean when I talk about the systemic circulation? The rest of the body. Everything else besides the lungs. What's the definition of the arterial circulation? It's just, you know what an artery is. What's the how do we define an artery? So there is that is the statement that is almost, but not quite right. So you think about arterial blood as being bright red, being oxygenated. There's an exception to that. Michael, do you know?
Takes blood away from the heart.
It takes blood away from the heart. So the blood that is going, so this blue blood going from the right ventricle to the lungs, it is deoxygenated because it hasn't gotten to the lungs yet. This is an artery, not a vein. So in the pulmonary circulation, deoxygenated blood is carried by arteries. But in the systemic circulation, Mackenzie was right. In the systemic circulation, oxygenated blood, in the systemic circulation, oxygenated blood is carried by arteries. The rule of thumb is what Michael said. If the tube is taking blood away from the heart, away from the heart, away from the heart, it's an artery. If the tube is taking blood back to the heart, back to the heart, back to the heart, it's a vein. So so in the systemic circulation, the arteries are carrying oxygenated blood and the veins are carrying deoxygenated blood. In the pulmonary circulation, it's the reverse. In the pulmonary circulation, the pulmonary artery is carrying deoxygenated blood and the pulmonary veins are carrying oxygenated blood. I'm sorry for that's just the way they're called. It's not just the way they're called, it's actually, this is an artery. It has the structure of an artery. Okay, so we've talked about the arterial and the venous, we've talked about all this stuff. So let's walk through the systemic circulation. So this is a broad overview. And then whether you like it or not, we're going to get down to the molecular, the cellular and molecular level. Right now we're talking about the broad overview of the system. Okay, systemic circulation. So we're talking about this part right here. The systemic circulation, let's just start, it's a circuit, it goes round and around, so we can start anywhere we want, we can start anywhere, but let's start with the left ventricle. The left ventricle is what pumps the blood to your body. The blood leaves the left ventricle and goes out the aorta. And we're going to see the aorta in lab today, or at least by the end of the night tonight, we'll see the aorta. The aorta divides into named systemic arteries. So, branching off of the aorta is the left subclavian artery that feeds into the brachial artery, axial, and then the brachial artery. And you may know that it's the brachial artery when the nurse or the physician puts on the blood pressure cuff. It's the brachial artery that they're reading the pressure of. So there are arteries that have names, and you're going to learn the names of these. It's in the coloring book, eventually, not right away. It's the names of the arteries that you do need to know are on the key concepts list. If the coloring book goes into more detail, this is just a general truth, if the coloring book goes into more detail and includes information that is not on the key concept list, it won't be on a test. The key concept list overlaid onto your coloring book is the detail that you need to know for an example. Okay, so systemic arteries. These are arteries, major arteries, that have names. And you're going to learn the names of many of them. Then those major arteries divide into much smaller, now we're getting into sort of microscopic, micro circulation. They divide up into tiny little arteries, but we don't call them tiny little arteries, we call them arterioles. So major arteries that have names divide up into thousands of arterioles that don't have names. They have a generic name together, arteriole. Those arterioles are what feed the capillaries. So you walked into this room knowing what an artery was, or almost knowing what an artery was. You walked into this room knowing what a capillary is. Most of you know capillaries are where blood exchange with the tissues happens. Those are the tiniest things. You may not have known the name arteriole, which is what it had in between an artery and the capillaries. Anyway, there's microcirculation. Capillaries is where nutrients are given out from the blood to and oxygen. Nutrients and oxygen are given from the blood to the organ. And waste products, carbon dioxide and other wastes, are absorbed by the circulation from the muscle or organ. Capillaries is where that exchange happens. The capillaries, the microscopic capillaries, then merge together to form tiny little veins, which we call, we don't call them tiny little veins, we call them venules, which merge to form systemic veins, which have names, which you will learn, which merge into the major vein of the body. There's two of them. One of them is called the inferior vena cava, which goes to the right atrium, which goes to the right ventricle. So that's the pathway of the systemic circulation. So, how many aortas do we have? One. How many named systemic arteries do we have? What's your guess? Scores? Maybe 100? Definitely more than 20. I'm not sure I could come up with 100 names. I'm pretty sure I couldn't come up with 100 names. Thousands of arterioles. How many capillary? How many of these tiniest tubes do you think we have? Thousands, millions, billions? Turns out it's billions. Billions of capillaries. Okay, so that's an interesting tidbit of information. There's a major, so this cartoon has a major defect. What part, so we've got the body here, you can imagine this being some skeletal muscle bed, maybe this is the digestive tract, maybe this is some other skeletal muscle bed. We've got the pulmonary circulation here, which we'll talk about more in a minute. What is missing? There's a major part of the systemic circulation that is missing. Kendra? The brain, right? So up here, we should have a piece of the systemic circulation feeding the brain. It's just, maybe this is supposed to represent the systemic circulation to the brain, but it's in the wrong place, way in the wrong place, okay? Just let's not forget systemic circulation to the brain. Okay, now let's talk about the pulmonary circulation. Right ventricle, which is where I ended the systemic, so right ventricle is the beginning of the pulmonary circulation. Beginning and end, that's a detail that we could debate, and it's not worth it. The right ventricle pumps blood into the pulmonary arteries, because we have more than one. We have one pulmonary trunk that divides into two pulmonary arteries. And then that divides into pulmonary arterioles, pulmonary capillaries, pulmonary venules, which then pulmonary venules merge together to form the pulmonary veins, which bring oxygenated blood back to the right atrium. No, got that wrong. Back to the left atrium. And then now the blood goes from the left atrium to the left ventricle, where we start this systemic circulation. But, so I described this in series, but this is happening at the same time. Blood is flowing to the lungs through the pulmonary circulation at the same time that blood is flowing through the arteries to the systemic circulation. It's happening simultaneously. Okay, questions about the overview of the circulation? Yeah.
So is there not an equivalence to the pulmonary trunk on the vein side?
No, there isn't.
Okay, so multiple veins are.
Multiple
connected
pulmonary
to the
veins will go from the lungs to the heart. 4. We'll see them in lab by the end of the night tonight. I don't talk a lot about blood, but obviously, so we're not going to talk about white blood cells and red blood cells and all the others, types of blood cells. That's not. There's a lot of things I don't talk about in this course, right? I mean, there's whole organ systems, like the skin is an organ system that we don't cover in this course. Why? Because I'd like to go deeper into some of the into some of the systems that I think are more interesting than others. So I don't go into the blood as a system, but it is a system. I don't go into the skin as a system. I don't really go into into bones as a system. So there are some things that you could say, wow, I'm getting shortchanged. Well, I don't think you're getting shortchanged because we're going into detail about the other systems. But let's just talk about blood for a minute. Your heart is pumping out blood, all the time. And so we talk about that output as cardiac output. And that output is in a volume per minute term. So you pump about 5 liters of blood per minute. 5 liters. Anybody have I should have brought my beaker. Anybody have a sense of how big a beaker it requires to pump a 5 liter beaker? Yeah, it's a bucket. 5 liters per minute is a bucket. We're not talking about a little beaker. We're talking about a bucket beaker. That's what 5 liters looks like. And your heart is pumping that every minute. And that blood is going to various parts of your body. So, for example, your brain is getting about 13% of that 5 liters every minute. And your kidneys are getting about 20% of that blood every minute. And your abdominal organs are getting more after a meal, less between meals, but getting on average about 24% of that blood every minute. Cardiac output is going to all the systems of your body. So here's a semi-trick question. What percent, so what's not covered here, what percent of the blood flow goes to the pulmonary circulation? What's the number? 100%. Because all of your blood, that we're not talking about a fraction of the systemic circulation, we're talking about the pulmonary circulation, which is different. 100% of your cardiac output goes to your lungs on the right side of the heart. 20% of the cardiac output on the left side of the heart goes to skeletal muscle. Okay? This is a trick question. First-year medical students often get tripped up by this question because they've memorized all these numbers. They've memorized 20%, they've memorized 9% going to the skin. They've memorized the numbers. But then you ask them a question like, okay, what's the answer to a question that you didn't memorize? Just like, but you got it. 100% of the cardiac output goes to the pulmonary circulation. So we talked about cardiac output. Now let's talk about where the blood is at any given time. Blood, the volume of the blood in the circulation at any moment in time. So on the arterial, on the systemic arterial side, not the pulmonary arterial side, but the systemic arterial side, left arterial side. Okay? At any given time, about a quarter of your blood is in the left systemic circulation. A quarter of the systemic blood, not the pulmonary blood, a quarter of the systemic blood is in the arterial side. And I call it the high pressure side because the left ventricle, let me just clarify something, the left ventricle is what generates the force and the pressure that pumps blood throughout your entire body, from your brain down to your toes. It generates a lot of pressure in order to get that flow. So we call it the left systemic side is the high pressure side. The high pressure necessary to pump the blood through all those tiny little capillaries. So if a quarter of the blood is in the high pressure side, that means 3/4 of your blood is in the low pressure venous returning to the heart side. So at any given time, most of your blood, 3/4 of it, is sort of in the slow-moving, low-pressure return to the heart side. So that's an interesting fact, that only about a quarter of it is in the high-pressure, flowing vigorously side. And so will I require you to memorize these numbers? No, but I will not. Let me be clear. I will not require you to memorize these numbers. I won't require you to memorize these numbers. That's for medical students. I do want you to understand the concept here is that cardiac output is divided into, the systemic cardiac output is divided into the various systemic organs that are demanding blood. The brain, animals, the brain, the heart, the skin, the kidneys, your digestive tract. And I want you to know the concept that less blood, only about a quarter of the blood, is on the high pressure side, the left side, and 3/4 of the blood is on the low pressure venous side. And some of it is in the heart itself, some of it is in arteries, and some of it is in arterioles and capillaries. Okay? You don't need to know the numbers, it's just this is supposed to add up to 25%. So only about 77% of your blood total is in the capillaries or the arterioles at any given time. Most of it, right there, that's the biggest number. Most of the blood in your body at any given time is in veins or small, tiny little veins called veins. Okay, so that's just an interesting bit of information. So here is what organ is this? That's the heart. So what's the name of this circulation? You've heard it before. It begins with a C. It's not the cardiac, we don't call it the cardiac circulation. Coronary. Coronary circulation. So here is and how was this made? Well, they just injected into the blood a dye that made it block X-rays so that you could see it in an X-ray. So this is an artery. It's got a name. It's the left. I think it's the left ascending. Not ascending, because D stands for descending. Left. I can't remember the name of this blood vessel, but it's a artery that's dividing into two other arteries that probably have names. That is now dividing into arterioles, and then the tiny, these tiny little things, capillaries, right? Your heart is a muscle. It not only is it pumping the blood, but it's receiving blood through the coronary circulation in order to provide the oxygen and nutrients that the muscle itself needs to work, and remove the carbon dioxide and metabolite products that we want to get rid of in the heart. The thing that gets confused, many people out there, you may have walked into this room thinking that the heart got its nutrients and oxygen from the blood that's flowing through it. The blood that's flowing through, this is the oxygenated blood that's flowing through it. But this is not, this blood here that's flowing through the heart is not providing the heart muscle itself with anything. It's the coronary circulation. So branching off of the aorta is this major artery that is feeding the blood to the heart itself. It's one of the circulations. Okay? So what is this? So I'm going to walk through this graph carefully because you need to know this information for one of the problem set questions. So this is plotting an area. And what's the formula for the area of a cylinder, for the cross-sectional area? So here is a artery. In cross-section, what is the name of that this is going back to elementary school. What's the name of the thing that this is in cross-section? A circle. You look it down the tube, it's a circle. What is the cross? I'm doing this because every year there's, somebody, what do you mean by cross-sectional area? I need to walk through this, and I try to make it as straightforward as I can, but I don't mean to insult you by asking you what is a circle, but let's just be clear. So what is the formula for the cross-sectional area of a artery? Pi r squared, where r is the radius of the tube. So pi r squared is the area of a cylinder, cross-sectional area of a cylinder. Not the surface area, but the cross-sectional area. Okay, so here we are. What tube are we in right here? It looks like it's aligned with the figure, but it's not really, it's only kind of. What tube is at the beginning of the systemic circulation? The aorta. How big is the aorta? Show me your estimate of the diameter, of the size of the aorta. It's about an inch. 2 centimeters in diameter. 1 point something in radius. Pi r squared, where r is 1 point something, is 4 square centimeters. So the surface area, the cross-sectional area of the aorta is 4 square centimeters. Where are we here? We're in capillaries here. How is it possible that the cross-sectional area of the capillaries is 3,000 square centimeters when the cross-sectional area of the aorta is only 4 square centimeters? Now,
There's so many of them, the total.
so many of them. What is that number? Billions of them. Billions, even though each one only has a cross-sectional area of maybe 10 to the minus 4th square centimeters. We got a billion of them, and so the total cross-sectional area, you add them up together, because they're all, one tube is feeding into multiple tubes. The cross-sectional area here, this is a bad example, but the cross-sectional area here is something, and the sum of these cross-sectional areas is greater, and in this system is a lot greater. So the total cross-sectional area of the capillaries is 3,000 square centimeters. You're going to need to use those numbers. Take the information from this graph to answer question 6 on the problem set. And I'm happy to help parse the questions if you need help. And you'll see when you solve the problem that the rate of flow in the microcirculation is very slow. And the rate of flow in the aorta is pretty fast. You'll see that when you solve the problem. You'll see so what I want you to do, I'm not going to give you the answers. I want you to solve the problem, but then I want you to think about, why did Bob ask me to do this silly geometry question? Well, it's because we get to some physiology there. I'm also asking you to use some of your quantitative skills. Okay, so I think let's just walk through some of the names. Let's spend 5 minutes walking through some names, so some anatomy, some cardiac anatomy, because it will become more informative. It's more informative if we talk about it sort of in theory with a simple cartoon, and then when we see it in real life, so to speak, when we see the real heart, the pig heart or the human heart, the name, it won't be the first time you'll be hearing at least some of those names. So let's start here with the left ventricle. Here is the left atrium. So where is this blood coming from? The lungs, and it's coming from the pulmonary veins. And it flows from the left atrium into the left ventricle through a valve called the bicuspid valve, or the left atrioventricular valve, or the bicuspid valve. And when this muscle is relaxed, blood is flowing through the open valve, but it is not flowing backward here because this valve, which is the aortic semilunar valve, is closed. And we'll talk about what opens and closes valves next week. Okay, I'm just introducing some names here. So blood flows through the bicuspid valve into the left ventricle. There are some structures inside the ventricle. Let's look at it for real in that. So here, we don't even need to make it move, but this is an actual heart. And you can see inside the chamber there are some structures there. So the cartoon that was up there in a moment was cartooning some real things, okay? And you'll see those real things by the end of the night tonight. So for example, these fibers that connect the inside wall of the left ventricle to the left AV valve, or the bicuspid valve, these fibers have a name, and it's only named over here, but it's got the same name. The chordae tendinei on the left and the right side. Chordae means strings or chords. Tendinei means tendon, like tendons, only they're not tendons. Chordae tendinei, the colloquial name for them is heartstrings. So when you hear the phrase, oh, that really pulls on my heartstrings, something that's emotional, it's actually got some cardiac anatomy behind it. Chordae tendinei are connected to the inside of the wall of the ventricles, both the left and the right, by papillary muscle. So we'll see papillary muscle. Papillary muscle is a specialized cardiac muscle that connects chordae tendineae to, and the chordae tendineae are connected to valves. Okay? Left side, left ventricle. The left ventricle, the muscle, the meat of the heart itself is called myocardium. On the right side, okay, so let's follow blood flow. From the lungs, left atrium, left bicuspid valve, left ventricle, and then when the left ventricle contracts, it pumps the blood out into the aorta through the aortic semilunar valve. The aorta splits into 3 major arteries. This one is called the brachiocephalic artery. This one is called the left carotid, the left internal carotid artery, and this one is the left subclavian. You'll get these as you work with the coloring book. Okay, so the blood goes to the body. Now it comes back, goes to the body, gas exchange, nutrient exchange, into the veins, returns to the heart through the inferior and superior vena cava. You'll see those in lab today. Well, some of you will see them at the end of the day and by 9 o'clock or 9:30 tonight. That returning blood goes into the right atrium, and it flows through the right AV valve, which has a name called the tricuspid valve, into the right ventricle. The right ventricle has papillary muscle, the right ventricle has chordae tendineae. When the right ventricle contracts, it pumps the blood out into the pulmonary artery, pulmonary trunk, and pulmonary arteries through the pulmonary semilunar valve. Again, I was just using the words so that you're not hearing them for the first time as you're struggling with seeing the actual heart. So that when you're seeing the actual heart, whether it's a pig heart or the human heart, you're able to map what you just heard onto what you're seeing in 3 dimensions. Are there questions about this anatomy? I think everything on this slide, maybe with the exception of septum, everything on this slide would be fair game on an anatomy question, either on a quiz or on anything. Questions? Yes.
Could you say again what AV stands for?
Atrioventricular. Yes.
How do you identify papillary muscle when you're actually doing the
It's like, you know it when you see it. It looks like, so papillary means nipple-like. It looks like a nipple, and it's connected to chordae tendineae. You'll see it inside the heart, and you'll see it as this big long nipple connected to chordae tendineae. You can't miss it. You can't miss it.